And chapter 10 is talking about how organisms reproduce. And we know that organisms reproduce using, most organisms use two cells that are called gametes. And we generally think of them in animals as sperm and egg. Plants, they may be different. It could be pollen and egg, and some other organisms, they have different names for them. But they are all gametes. And we know some things about gametes now that we'll get into when we talk about meiosis, how we make those gametes. But I want to start with actually the second section of chapter 10, which was Mendel's work. So Mendel was a monk, a priest, but that's not what he's remembered for. He's remembered for his work as a gardener. Mendel wanted to know what people have wanted to know for a very long time, and still really want to know, is how to predict offspring. So people have bred animals, people have bred dogs to get the best breeds they want, to get the characteristics they want in a breed. People have bred cattle to try to get the best characteristics that they can. But it all seemed to be, up until Mendel's time, a random, almost random, not really random, because you knew some idea what you would get, but it was always thought to be a blending, that you'd get some combination of both parents in the offspring, and that combination tended to look in between the two parents, not all one or all the other, and not in a predictable way. Mendel was lucky in that he chose to look at pea plants, and pea plants were lucky for him. First of all, you may not think of it this way, but pea plants reproduce sexually. They have gametes. That's all that means. In order to make a new pea plant, you need a gamete from one pea plant to combine with a gamete from another pea plant. So they reproduce sexually, just like most of the organisms, dogs, cats, things we try to breed. But they were a little simpler model. Another reason they were important for him is he could control the breeding. He made sure he knew where each of those gametes came from. And for 7 traits, there were only 2 phenotypes. Their plants were either tall or short. There was no in between. Flowers are either on the end of the vine or sides of the vine. And there were 5 other traits that were just like that. There were only 2 options. There was no combination. There were no middle-sized pea plants. There were no pea plants with flowers on the end and on the side. There were no green peas. There were green peas and there were yellow peas, but there was no in-between green and yellow color. So he had these plants that reproduce sexually. He could control the breeding, and there were only 2 phenotypes for 7 different traits. And remember, he called those true breeding plants. Which we now know means they were homozygous for those alleles. So with his pea plants, he decided to cross them, and he crossed a tall plant with a short plant, and this was a tall true breeder, this was a short true breeder. Now initially he expected to get medium-sized plants, but when he did this cross, he got all tall plants. So then he decided to take these tall plants, which he called hybrids, because they were a combination of a true breeding tall and a true breeding short. And when he crossed two, put a little H here, two tall hybrids, he got 3 tall and 1 short plant on average. So that was the gist of his experiment. That's what he did, and he did it with thousands of pea plants. He worked it all out. And what he got from this information was, first of all, that traits like height, flower location, color of peas, color of pods, shape of the peas, traits are controlled by 2 factors. And he had no idea about genetics, about genes, about what those factors were. He just knew there were 2 physical factors in an adult plant that controlled how tall it was, whether it was tall or short. One of those factors came from the mother, and one of those factors came from the father. So traits, at least the 7 he looked at, were controlled by 2 different factors. He also, and in his case, for all 7 things he looked at, this is true, but it's not true for all genes. He said that those 2 factors came in 2 forms: dominant and recessive. So in this case, since all the offspring, all the hybrids were tall, the tall was the dominant and short was recessive. Now, so if you have 2 factors that come in 2 different forms, there are 3 different combinations of those you can have. You could have 2 factors that were the dominant. You could have both of those factors be the recessive, or you could have one of those factors be the dominant and one be the recessive. This would give you a tall plant. This would give you a tall hybrid plant. And this would give you a short plant. And because those are the same, they are homozygous. They are true breeding tall and true breeding short. And this one, not just a hybrid, it's a heterozygous plant. So taking all that into account, he came up first with the law of segregation. And the law of segregation says that those 2 factors in an organism must separate in gamete formation. So those two factors that control things like how tall a pea plant is have to separate from one another in the making of gametes. A gamete will only have one copy of that factor. And now we know that those factors are genes, and the different forms of those factors are alleles. So there is one gene that has two copies that controls how tall a pea plant is. There are two possible alleles for that gene, a tall allele and a short allele. And then you could have two tall alleles and be true breeding or homozygous tall. You could have two short alleles or be true breeding short pea plant. Or you could be a hybrid. You could be a tall allele and a short allele, be heterozygous. If that's the case, the shortness is still there. It's just hidden by the presence of the tallness. So you get a tall phenotype for that plant. The second thing that Mendel came up with, the second law, is the law of independent assortment. And the law of independent assortment you can't get from doing just one cross at one trait. The law of independent assortment deals with two or more traits. And that says that genes for one trait don't care about the genes for another trait. So that if you were looking at tall and the color of the peas, you're going to get all possible combinations of alleles in gametes for the tall genes, and it will be irrespective of how the color of the pea genes go. So what that means for us is when we are doing crosses, we have to take into account all possible combinations of genes. So if we have gene X, which has the alleles X and Y, and we have gene B, which has the alleles big B, little b, this X could end up in a gamete with this B. This X could also end up in a gamete with this B. This Y could end up in a gamete with either this B or this B. So all possible combinations of X's and B's are possible because of the law of independent assortment. The law of segregation says that these two X's can't be, this XY can't be in the same gamete. These two B's can't be in the same gamete. But the law of independent assortment says that they can be with either one of these, and these can be with either one of those. So those are the two laws that Mendel came up with by doing some very simple yet mathematically complex work with pea plants. And after Mendel's work was lost, and just before it was rediscovered, science came up with some new information. The first of which, or most important for us, is the process of meiosis. And that's 2 rounds of division with 1 copying step. So one of the problems with combining cells from two different organisms, or two different members of a species to make a new species, has to do with chromosome number. So humans have 23 chromosomes. So if two humans joined two regular cells, the new cell would have 46 chromosomes instead of 23. And 46 chromosomes is not the number of chromosomes in a human, so that stem cell you would create that way would not be a human stem cell. So they had to figure out exactly how do we get a cell that instead of having two copies of each only had one copy of each. And that was worked out when the steps of meiosis were worked out. So here is a human gamete stem cell. That stem cell has 23 pairs of chromosomes, 2 copies of 1, 2 copies of 2, all the way down to the 23rd. That stem cell is going to go through G1, S, and G2. What's important for us though right now is the S phase. So it's going to make copies of the chromosomes. So after S, there will be (43) pairs of chromosomes. There will be 4 copies of number 1, 4 copies of number 2, 4 copies of number 3, and so on. Those 4 copies are 1 copy of the maternal and 1 copy of the paternal. Don't forget though, well, we'll cover it about crossing over, which we'll get to now. So those after S and after G2, let's call this a G2 chromosome, we go through prophase. And this is meiosis prophase. And in meiosis prophase, we get the formation of tetrads. So those 4 copies of number 1 are going to be, whoops, those 4 copies of number 1 are going to be tightly crossed and tangled. And that's going to lead to a crossing over phenomenon. And that crossing over phenomenon gives us more variability. So in meiosis prophase 1, we get tetrads forming. Those tetrads are each one of those 23 pairs, but are now 43 pairs, are all together. So all 4 copies of 1 are together, all 4 copies of 2, all 4 copies of 3, all the way down to the 23rd. They're tangled, crossed over, and the easiest way to fix them is to cut them and tie them back together. So we end up with 4 unique tetrads now, 4 unique chromatids now. We don't have a chromatid that this organism's mother gave to it. You have a chromatid with pieces of DNA from this organism's mother and this organism's father. You get 4 different new chromatids, new combinations of chromatids. From prophase 1, we go to metaphase 1. Metaphase 1 is just like metaphase in meiosis, except tetrads line up. So now these 4 new chromatids, all the chromatids from number 1 line up together, all the chromatids number 2 line up together. In anaphase 1, In anaphase 1, the tetrads are going to move away from each other. So 2 of the copies of number 1 will move to the left, and 2 of the copies of number 2 will move to the right. And that is because those tetrads are held by centromeres. 2 copies are held by centromeres. So the centromeres are not going to split. And that will be followed by telophase and telophase and cytokinesis 1, same as anaphase 1, metaphase 1, prophase 1, which will give you 2 cells with 1/2 tetrads in each of those 2 cells. They will then go through meiosis 2 or 2 depending on which you want, and that will give you a division that is just like mitosis, because 1/2 of a tetrad is 2 sister chromatids joined by a centromere. So that means that meiosis 2 looks just like mitosis. And that means you get 4 cells. Each one of those 4 cells contains 1/4 of a tetrad, and 1/4 of a tetrad is 1 chromatid, 1 of the 23 pairs. So instead of 2 copies of number 1 that you had in the stem cell, there is 1 copy of a number 1 in the gamete. And these are gametes, and those would be sperm or egg. So that process of meiosis explains the law of segregation. If you have 2 copies of number 1 in a stem cell, in a gamete you can only have 1 copy of number 1. So those 2 copies of number 1 have to separate from one another. That's the law of segregation. If you have 4 completely different gametes, that is because of independent assortment. So crossing over in the formation of tetrads explains the law of independent assortment. But unfortunately, or fortunately I guess, we do know that sometimes that law of independent assortment looks like it got violated. And that happens when genes are linked. So when you have gene linkage, your chromatid has 2 genes that are very close together. And if the 2 genes are very close together, it's less likely that they will cross over. So that allows us to use the percentage of crossing over to map chromosomes. The more often you get crossing over between 2 genes, the less linked they are. The less often you get crossing over, the more often the 2 genes are, 2 alleles are inherited together, the closer they are on the same chromosome. So it allows us to make a map of chromosomes. All right, I'm going to stop there. Tomorrow I'm going to post a review that's based on the summative assessment that will be up soon for this section, and then by the end of the week we should be wrapping up this chapter. Send me any questions, and I would also like to say that on MobyMax there is are lessons that will help you with meiosis and sexual reproduction if you're having trouble following me. They're optional lessons, but they are there and they're pretty good. So take advantage of them.